Well, hello everyone. Thank you for the opportunity to speak today, and thanks to everyone who tuned in. My name is Hassan. I'm an assistant scientist at the Miami Project to Cure Paralysis in the lab of Drs. John Bixby and Ben Sleman. Research in the Miami Project revolves around spinal cord injury, and our lab focuses specifically on the problem of axonal regeneration in the CNS. Today I will present some of the work that I've done to identify small molecules that can induce CNS axon regeneration. A typical exercise in drug discovery involves finding a biological target that can be engaged in order to induce a desirable outcome such as resolution of a disease phenotype. The idea is that you have a biological network that has become deregulated for some known or unknown reason. You identify a molecular target that regulates the network. You engage this target with a drug, for example, a small molecule, and you restore some level of normalcy for the network. Common wisdom says that off-targets are implicated in adverse effects and toxicity. Therefore, drugs should be maximally selective for the intended target. A missing part of this story, however, is that engaging additional targets can, in certain cases, potentiate the desirable effect. This phenomenon is known as beneficial poly-pharmacology, and it's quickly gaining popularity as more and more drugs are revealed to work through multiple targets. And here are some examples from kinase inhibitor drugs that appear to work through multiple kinase targets, the first of which was, of course, imatinib. The ultimate goal of systems biology is to generate high-resolution models that will enable us to predict which targets or target combinations need to be perturbed in order to efficiently induce a change in the typically robust biological networks that tend to resist perturbation very well. Towards that goal, the field is currently working to overcome some pervasive challenges, which include biological level complexity, where modeling the functional state of the cell based on genetic level data may lack critical functional information at one or more levels. Time-dependent complexity, where accurate modeling of processes that are nonlinear in the time dimension is constrained by limited time sampling. The difficulties that are inherent to large data analyses, including finding the right balance between having a very wide scope while also focusing on the most relevant molecular events. And finally, there's the more general challenge of drugging a new target where the majority of the proteome is predicted to be undruggable. Meanwhile, two major approaches dominate drug discovery. We have target-based screening, which has come to popularity in the genomics era with the thinking that a single disease-modifying gene can be found for a given therapeutic application. Here, compounds are tested in a biochemical assay with a single target, and compounds are prioritized based on the readout on this target. Then we have phenotypic screening, where compounds are tested on whole cells or tissue and prioritized based on the phenotypic readout. So in phenotypic screening, you don't require a target hypothesis. And so you're able to discover highly effective compounds, including those that exploit beneficial polypharmacology. However, the targets are often difficult to identify and frequently remain unknown. And this makes lead optimization difficult. On the other hand, lead optimization is quite straightforward in target-based screening, but the technique is agnostic about off-target effects, be they bad or good. So the question on my mind was, can we combine the two techniques to detect consistent target behavior? In this scenario, we would screen compounds in a phenotypic assay and pick out our hits based on some desirable phenotypic readout we decide. Then we would screen those hits in a series of target-based assays, let's say for enzymatic inhibition. And we would look for biochemical activity patterns that strongly correlate with the desired phenotypic outcome. In this trivial example, you can see that proteins 10 and 18 may be possible candidates. By adding in data for non-hits, we can further refine our predictions. For example, here we can now see that number 18 is not in fact correlated with the hits, leaving number 10 as the only target candidate. By extension, we can also make predictions about proteins whose engagement correlates with the unwanted phenotype, hence anti-targets. In this context, the concept of anti-target is extended beyond toxicity and adverse effects to include negation of biological efficacy. So for the proof of principle study, we directed our attention to kinases for several reasons, including the fact that over 500 kinases are coded by the human genome. They regulate most cellular processes. They are readily druggable. And given these features, they have become very attractive drug targets, which has resulted in the production of a tremendous number of small molecule inhibitors, as well as commercial kinase activity panels that can easily profile compounds with hundreds of kinases. A final important attribute is the homology in the catalytic kinase domains gives rise to inhibitor promiscuity, where a single molecule can potently inhibit multiple kinases. This could be a bad thing in that it makes it very difficult to design hyperselective inhibitors, but it can also be a good thing because it creates a platform for discovering and exploiting intentional polypharmacology. So going back to our biological problem, axons descending from the brain through the spinal cord regenerate very poorly after traumatic injury. This is thought to result from two main reasons. First, a programmed age-dependent loss of regenerative capacity in CNS neurons. And two, the presence of extrinsic inhibitory molecules in the CNS microenvironment. The combination of these two factors leads to this poor regenerative capacity, which ultimately results in paralysis, unfortunately. And so this appears like a situation where engaging multiple targets may be required to induce a robust biological response. In fact, most approaches that focused on individual targets or even individual pathways have not produced strong regeneration. We began with a phenotypic screening assay that uses mammalian CNS neurons isolated from embryonic rat brains. The assay measures the effect of screened compounds on neurite outgrowth. So to describe the assay briefly, neurons are dissociated after being dissected out of the brains and plated at low density on poly-D lysine. They are treated with kinase inhibitors at different doses, cultured for 2 days, then fixed and immunostained. After that, plates are imaged and traced using an automated Celomics robot and microscope, and the data is finally analyzed using MATLAB. The assay gathers tens of parameters on individual neurons, being a high content analysis. For this application, however, we focused on neurite outgrowth, which we quantified relative to DMSO controls and represent as percent NTL. Here I'm showing you examples of traced fields taken from cultures of control neurons, which as you can see have a hard time extending long neurites on PDL. Then there's neurons treated with a neurite outgrowth promoter, and you can visually see a strong effect on neurite outgrowth. And then there's neurons that have been treated with a neurite outgrowth inhibitor. So using this pipeline, I screened over 1,600 kinase inhibitors that were obtained either commercially from commercial suppliers or acquired via collaboration from GlaxoSmithKline, Roche, the Finland Institute of Medicine, or SGC. Compounds were screened at 5 doses, and hits were defined as compounds that induced an increase of neurite outgrowth by 30% or more over controls in 2 or more independent screens. The false discovery rate for the assay was about 7%, and the Z-prime score ranged between 0.7 and 0.8, meaning that it was quite robust. The next step was to select the compounds that would be most informative for the target deconvolution exercise. Here we employed a technique called stratification. Basically, if you have 2 sets of elements that are continuous and you want to understand the features that make these sets different, you remove the elements that are at the interface so that you accentuate the feature differences between the sets. In this particular case, we removed compounds whose neurite outgrowth effect hovered within 1 standard deviation plus or minus from the hit selection cutoff. And now we ended up with 2 sets of stratified compounds, the stratified hits and the stratified non-hits. Then we obtained kinase activity profiling data for a total of 265 compounds, which were a combination of stratified hits and stratified non-hits against a total of 190 kinases. The majority of this data came from our collaborators at GSK, and we added profiles for about 20 hits to enrich the analysis and accentuate the features that reflect targets. We used the maximum relevance algorithm to rank the kinases by their information content or relevance to the 2 compound classes, the compound classes being hits and non-hits. So here are 2 fairly obvious examples of kinases with high relevance as suggested by their inhibition bias by either the hit class or the non-hit class. So you see the one to the left. It's frequently inhibited by compounds that are hits, but not so by ones that are not, and the inverse is true of the other kinase. And here are 2 examples of kinases that show low relevance. One, because it's simply not being inhibited by any of the compounds, so it has no information relevant to the phenotype in this context. And the other one because it appears to have a uniform inhibition pattern in both compound classes. So this relevance parameter was quantified using this equation, which calculates the joint and marginal activity probabilities for each kinase with each one of the 2 compound classes. Using this equation, we ranked the kinases in order of decreasing relevance, and the top 50 were selected for subsequent analysis. As you can see here, the very clear case of the most relevant kinase, where you see that a lot of the activity, actually all of the activity clusters in the hits and none of the non-hits inhibit this kinase. Next, we gave this data to a support vector machine, which is a type of machine learning algorithm, and prompted it to perform a 10-fold cross-validation in order to check how well it can predict whether a compound is going to be a hit or a non-hit in the phenotypic assay based on its activity against the 50 relevant kinases. Encouragingly, the accuracy came at 80%, which is well over random guessing. It wasn't 100%, but you don't expect 100% when you've only sampled 190 possible targets out of everything out there. So next we wanted to prune down those 50 kinases to the smallest set that could produce high classification accuracy. In other words, we wanted to narrow down things to a small set of targets that have the largest influence on the phenotypic outcome. So we prompted the SVM to remove kinases one by one and track the prediction performance. If removing a kinase degraded the accuracy, the kinase was deemed influential and the SVM had to put it back in. Otherwise, the SVM could drop the kinase and move to test the next one. Since I was mostly interested in finding targets, I devised a simple differential prediction metric to track the performance. It basically works by maximizing the algebraic separation between sensitivity and error in this exercise. And here is a sample output from the overall analysis, which is abbreviated MRSVM for maximum relevance support vector machine. So now things may be more obvious that you can detect them visually, and you can see that the activities for the hits cluster in one end and the non-hits in the other, and one might start making hypotheses that maybe the kinases to the left are behaving as candidate targets while the ones to the right are behaving as candidate anti-targets. However, there's a caveat. I mentioned earlier that the homology between kinases gives rise to inhibitor promiscuity. And when two kinases are very closely related, they can have near identical inhibition patterns. What this means to the algorithm is that they will be treated as equals in the analysis, even if only one of them is biologically relevant to the phenotype that we're after. To overcome this, we clustered the kinome into pharmacologically linked groups. Optimally, these groups would be inferred from compound inhibition data because sequence data cannot always capture binding behavior. Nevertheless, in the case of kinases with their shared ancestry, estimating linkage by sequence similarity works to a good approximation. So we rolled up these kinases within the kinome by sequence similarity of their catalytic kinase domains. The assumption being the closer the sequences, the more likely they are to bind the same compounds. We also informed the grouping with the inhibition data we had available, which allowed us to combine groups that have similar binding patterns despite having divergent sequences. For this step, we used the pharmacology interaction strength parameter as described by Metz et al. This left us with 305 groups of kinases that are expected to show high pharmacological linkage. So in order to test the actual biological relevance of our predicted targets, we needed to assess the contribution of each predicted kinase as well as all its linked relatives to the cellular phenotype. In this example, we would have to test kinase 15, 132, 21, and 19 to be able to make a statement about this group. For this, we used siRNA-mediated knockdown, where we knocked down kinases in the top predicted groups to see how their behavior relates to their inhibition data. In this figure, the kinases are arranged from left to right, as you saw before, where the ones to the left are the ones that show higher inhibition bias by hits, while those to the right are the ones that show higher inhibition bias by the non-hits. The dashed lines indicate group boundaries, so kinases within dashed lines are members of the same group. Given the caveat of relating knockdown and pharmacological inhibition data, we did not expect a strict correlation in the result. However, we did want to see an overall pattern where knockdown of candidate target kinases results in neurite outgrowth induction, while knockdown of candidate anti-targets would have the opposite effect. And we do see that. Additionally, we noticed that groups whose members behaved concordantly or in a similar manner had the highest inhibition bias in either direction, and thus ended up on either the far left if they were targets or the far right if they were anti-targets. While groups with discordant behavior clustered around the middle. And you can imagine what might be the reason for this, because if these groups are made of pharmacologically linked kinases, a compound that inhibits one member within a group is likely to inhibit other members. So you're frequently getting an averaged readout from the entire group. If the group members are acting in the same direction, the readout is going to be shifted to the left if they're all acting as targets, or to the right if they're all acting as anti-targets. But if they're acting against each other, things are going to hover around the middle. Since the purpose was to focus on kinases that had the most robust influence on phenotype, we picked the 10 groups with the highest inhibition bias. This left us with 7 target groups and 3 anti-target groups. And here are the 10 groups, each represented by one of their members. So again, you can see the clear pattern of inhibition of targets correlating with hit behavior and inhibition of anti-target correlating with non-hit behavior. A worthwhile observation is that 2 of the targets, ROC and PKC, were already known to mediate axon growth inhibition by CNS matrix molecules. As 6 kinases have also been known to inhibit the intrinsic growth capacity of CNS neurons. While CDK5 and P38-MAPK were already known to be required for axon growth. So this was a nice additional piece of validation for the approach from known literature. In this figure, the compounds are arranged from top to bottom by their maximum effect on neurite outgrowth. So a striking observation was this top compound, which had very strong poly-pharmacology against 5 out of the 7 targets and no activity towards the anti-targets. Interestingly, this compound had by far the highest effect on neurite outgrowth in the cell-based assay. So we proceeded to test the compound in vivo and tried it in a pyramidotomy model in collaboration with the J. Lee lab here at the Miami project. In this experiment, the axons of the corticospinal tract are transected at one side of the mouse's brainstem, while the cell bodies sending the axons on the opposite side of the brain are transduced with a GFP labeling virus. The transected axons will degenerate, while the GFP labeled axons will sprout into the now empty region, and they can be counted. We implanted an osmotic pump into these mice and allowed the drug to be infused out of the pump into the CSF fluid. This went on for 28 days, after which time the mice were sacrificed and their tissue fixed and analyzed. As you can see from the panel to the right, the mice that were infused with the compound had double the amount of axon sprouting in B and D compared to those that were just infused with the vehicle in A and C. So given this encouraging data, we next decided to test the compound in a more stringent animal model. We did this in collaboration with the Xiaoming Zhu lab at Indiana University. In this experiment, the dorsal upper side of the the mouse spinal cord is lesioned to transect the CST axon running down from the brain along the length of the spinal cord. And the animals receive a single injection of compound or vehicle into their brain within the cortical region. As you expect in the controls, we do not see any regeneration. In fact, we see a gap where the lesion is, and we see that the CST axons are retracting away from the lesion site. In the compound-treated group, we saw a striking amount of regeneration. The CST axons grew through the lesion and down caudally from the lesion site. There was also an enormous amount of sprouting into the gray matter, which according to our collaborators was a very atypical result. And here is the quantification of the results from 7 animals in each group with a representative reconstruction of the response in control and compound treated animals. So you can see how extensive the regeneration is in the treated animals and also the amount of sprouting versus the control animals. And here you can see quantified the amount of sprouting versus also the amount of regeneration in the treatment group versus the controls. So we're currently gearing up to do some medicinal chemistry on the lead compound. However, since then, our collaborators at GSK have profiled additional hits from our phenotypic assay with the 10 representative targets and anti-targets. And we found that several of our highly active hits actually shared the poly-pharmacology profile with our lead compound. It's important to note that these hits have very diverse chemistries. This further suggests that the effect that we're seeing on neurite outgrowth is being induced by the poly-pharmacology that we're observing rather than some scaffold-specific off-target effect. So you can probably imagine how useful this is for a medchem project, that you can do this kind of scaffold hopping without losing the critical activity, both in terms of biochemical activity and phenotypic activity. Another interesting observation that we made was that combining compounds with complementary polypharmacology appeared to strongly potentiate their phenotypic effects. Here you see 2 hits which between the 2 of them inhibit 7 out of 7 targets and don't inhibit any of the anti-targets. When we tested these 2 hits in combination in the cell-based assay, we observed a very strong effect on neurite outgrowth. We had tried to find synergistic or additive combinations before by systematically exploring combinations of strong hits, but we were not able to produce anything of this magnitude. I tested the combination over a small dose range. I wanted to quantify the combination index of the two compounds, but unfortunately the effect was still maxed out at the lower concentration, preventing me from doing a fit. So I will have to repeat this at a lower dose range, which I'm doing right now. But it's still obvious the amount of synergy between these 2 compounds, or I mean, I can say additivity to be more conservative. So switching gears a little bit, we decided the applicability of the approach to cancer. Christer Winterberg, a collaborator at the Finland Institute of Medicine, had tested the same GSK kinase inhibitors that we tested in cancer viability assays. And Christer scores his compounds from the phenotypic screens using a drug sensitivity scoring system. So rather than recording the raw amount of cell death, the drug sensitivity score measures the selective cytotoxicity of each compound against the cancer cells versus some control cell. So using this data from an ERB plus breast cancer cell line, SKBR3, we ran the analysis, and sure enough, the ERB group was in the top identified targets. Interestingly, the pipeline was also able to pick out some kinases that were recently implicated in mediating the adaptive response of this cancer cell line to treatments that target the ERB receptor tyrosine kinases. Another collaborator at the University of Michigan, Max Wellner, had also tested these kinase inhibitors in a variety of cancer cell assays. So Matt has screened these compounds in 2D and 3D cultures with triple negative breast cancer cell lines. And we've run some analyses through the pipeline, and we're finding some interesting things where a kinase appears to behave as a target in the 3D but not the 2D assay. So we're right now in the process of following up on some of these results. Another interesting example emerged from a screen that Matt performed with a synovial sarcoma. We found a very robust anti-target in this cell line where inhibition of this kinase strongly induces proliferation in these cells. So we looked up 4 fairly selective inhibitors for this kinase, as you can see here from their inhibition profiles. And all of them strongly induce proliferation in these cells. Matt has RNA-seq data that further supports this observation, and he's currently performing additional validation of the anti-target using siRNA-mediated knockdown. And hopefully things will validate there as well. So this just seems like the kind of thing you'd want to know if you're designing a drug for that cancer. So to conclude, approaches that directly relate biochemical to biological data can efficiently identify biological targets. And indeed, several papers have recently appeared to describe such approaches, including the one in the August issue of ACS Chemical Biology in which we describe the current study. These semi-empirical methodologies can help bridge the gap between systems biology and classical drug discovery, and maybe provide a framework for guiding and refining those systems. Then there is the immediate utility in finding readily druggable targets, seeing that you're starting from assayable targets, and in being able to detect and exploit therapeutic poly-pharmacology. And finally, as comprehensive biochemical profiles become available for FDA-approved drugs, this kind of approach may be used to expedite drug repurposing. So in closing, I would like to thank everyone who has contributed to this work, particularly my mentors and now bosses, John Bixby and Vance Lemon, members of the Lembix Lab, and of course all our pharma and academia collaborators. And thank you all for listening, and I'll be happy to take your questions now.
Hello. I have a question for you.
Hi.
Hi, it's a very nice presentation. I'm wondering whether you have tried using cheminformatic approaches such as to compare their structural similarity and provide additional support for your analysis when you're trying to identify the drug candidates?
What kind of cheminformatic analysis are you thinking of?
Structural similarity.
So if I've tried to cluster the compounds by similarity, and if I found any similarities in the ones that were hits?
Mm-hmm. Or
yes.
If it's anti-hit, would there be distance? Their structural similarity would be much more different.
Right. So I have done that, and I found no correlation between structure and activity. but I did find correlation, as you saw, between activity against targets and the phenotypic outcome. So you can find a series of compounds that are similar within one scaffold. Some of them would be active and some of them would not be active, and the ones that would be active, you could see that they have activity against the targets. Or the inverse could be that the ones that are not active are actually engaging the anti-targets. So the simple answer is that there is no correlation by chemical scaffold. Does that answer the question?
Yeah. Thank you.
Any additional
So
questions?
for the 500 kinases, because you were from neuron to cancers, are those 500 kinases all present in the tissue? I assume they are all present in the tissue you study.
Mm-hmm. So
can you make any comments on
sure.
whether or not there's initial distributions of those kinases in different tissues? Because you say that you want to look at the drug candidate from the toxicity point of view. Mm-hmm. The off-target effect could be very important.
Right. So in the analysis, I excluded compounds that appear to have toxicity against the cells. So I'm not getting information about toxicity in the target deconvolution analysis. So if I include those, maybe I would be deconvolving targets that can contribute to toxicity. But to focus the study specifically on what's regulating neurite outgrowth, I have excluded toxic compounds and only included compounds that either promote neurite outgrowth, don't do anything to it, or inhibit neurite outgrowth without killing the cells. So in that respect, when I talk about anti-targets, I'm only talking about anti-targets for the phenotypic effect. But of course, there could be toxicity in anti-targets that we haven't looked into. So our lead compound would have to be passed through a toxicity panel to answer those questions, which we have not addressed yet.
Have you come across any research study done to understand gender differences in biological response? And in the assay people study, the people add estrogen or testosterone, try to make the phenotype more obvious, more detectable. Have you come across that kind of study?
I have not.
Thank you. Very nice presentation.
Thank you very much.
I have a quick question. So what was the assay that you use in testing kinase activity?
So this was the nanosyn assay, and it was an activity assay, not a binding assay. Do you want details about the assay?
Sure, if you could. So, yeah.
As I remember, it was an electrophoretic mobility shift. So the substrates are fluorescent, and after the reaction is terminated, they run through HPLC, and based on where the peaks appear, they know how much of it was reacted.
Okay. So is that something that's off the shelf, or it's kind of proprietary?
Yes.
Oh, so. Thanks.
I mean, they have that as a commercial service. That's how we profile some of our compounds.
Okay, cool.
Hassan, this is Stefan. Can you quickly comment, just for the folks who know links, on the concentration of the kinase profiling assay? What worked well, what didn't work so well?
Right. So I had, for the GSK compounds, or for the compounds that I used to run this analysis, I had profiling at 0.1 micromolar and 1 micromolar. And these two worked almost equally well, but the 0.1 micromolar worked better. So what do I mean by worked better? The algorithm was able to make better predictions from the 0.1 micromolar data about phenotypic effect than the 1 micromolar data. I also had another set of data that was collected at 10 micromolar, and that did not work very well. It appeared that the noise was too high, and the selectivities were just drowned, so the predictions were really degraded. I cannot say this with absolute confidence because the compounds were not the same, so it was a different library that was screened at 10 micromolar. But I also had another one that was screened at 0.5 micromolar through reaction biocorpse assay, and that also didn't work as well. So there I have two different sets of compounds, one at 0.5 and one at 10, and neither of them worked as well as the compounds when they were profiled at 0.1 or 1 micromolar.
Yeah.
So another point, in the phenotypic assay, I screened my compounds on the cells from 32 micromolar all the way up to 20 micromolar. Sorry, 32 nanomolar to 20 micromolar. Because the compounds were assayed in vitro with the kinases at 0.1 micromolar, I dropped the phenotypic assay concentration that was below that. So I was only relating in vitro kinase activity to phenotypic kinase activity at concentrations that are equivalent or higher.
Right, yeah.
So it would be, I don't know if it would be as meaningful if you have in vitro kinase data at 10 micromolar and you're trying to relate it to a phenotypic data at 1 micromolar.
Right. I mean, kinase inhibitors are typically much more active, right? So I think 0.1 makes sense,
Right. And you're dealing with issues of solubility, membrane permeability, and you have, there's a lot of things that you can't even account for. But as a first step, as a reasonable approach, you don't want to go too high with the concentration in vitro so that you can maintain as much relevance as possible to what's happening in the cells.
Yeah.
I just want to make one comment first. I thought it was also a very nice presentation. And the other thing is the idea of bringing two data sets, complementary data sets together is something that we also try at the DCIC for other data sets, and that's there is this power of making a prediction using one data set to predict the other. And also what it can be used for is to calibrate the pipelines that extract the final sort of signature from the data. So I'm not sure what type of methods you're using to, let's say, extract the features, the phenotypes from the imaging for the neurite outgrowth assay. But one way is to say, okay, the compounds can predict the outcome, and now I can play with the parameters to get a higher predictive model, a better predictive model if I tune the parameters of processing the images of the neurite outgrowth assay, and then you can do that on the other side. Let's say the neurite outgrowth assay is sort of like your gold standard, and then you can tune the parameters of the kinase inhibition assay processing pipeline. So just a suggestion. I'm not, overall I think it's a very nice complementary project that is use of very smart and use of big data, and it's sort of like the way to go forward with a lot of projects.
Thank you for your kind feedback and also for the question. And what you said is absolutely true. And we have thought about these things. For example, we acquire a lot of phenotypic data from these cells. And as I said, I narrowed it down to just one parameter. But the reason for doing that for now is because, one, it was a proof of principle. I didn't even know if this was going to work at all. So I just wanted to cluster phenotypes so that I can get more information out of the data. But breaking things down and trying to tweak the phenotype to see how the predictability will change from the data is definitely something interesting worth doing, and the inverse as well. And I'm happy that you picked up on the aspect of being able to use this empirical data, which is kind of like a majority vote from a lot of compounds, so that you can, pull out targets from the noise, and then you can go back and you can refine the big data and the big signatures that we're trying to work with, which would maybe help us narrow more on the molecular events that are relevant to what we're studying, and to find the nodes within the network where most of the perturbation or most of the network controllability is residing. Hello.
Yes, hi, this is Peter Hornbeck. I had a question trying to understand the effect of the knockdown on the candidate kinase targets and anti-targets on neurite outgrowth.
Mm-hmm.
So the ones that are positive there, for instance, on the left-hand side, that have large NTLs and large LNLs, or no, large, yeah. So those guys are the 1, 2, 3, 4. The fifth group in are the growth SGKs, rather the
GSK.
Axis kinases. And those are growth promoting. So is it, and you're inhibiting them, and you're getting a strong response in growth?
Yes. So you mean growth promoting in the context of cell volume and cell division, right? You're referring to S6 kinase role.
Yeah.
As so that's interesting, because it was a surprise for us at first as well, because we were thinking, okay, cell growth, axon growth, but it turns out that the two are not regulated in exactly the same way, and we're actually following up on that, and we found that inhibiting S6 kinases well, I can't give away too much because we are about to publish this right now, but this has been extensively validated with follow-up studies, and inhibition of these kinases does in fact lead to axon outgrowth, or neurite outgrowth at the least.
Terrific, very interesting.
Well, if there are no questions, maybe we should conclude. Thank you, Hassan. That was really fabulous. Thank you, everybody, for listening.
Good.
Thank you, everyone.